Welcome to our journey into the fascinating world of DNA. Deoxyribonucleic acid, or DNA, is the hereditary material found in almost all living organisms. It carries the instructions that guide the growth, development, functioning, and reproduction of every cell. In this video, we'll explore what DNA is, how it's structured, and why it's so essential to life. By the end, you'll have a clear understanding of how DNA shapes who we are and how it operates at the most fundamental level of biology. Let's begin by looking at the basics of DNA and its discovery. Story of DNA's discovery is a tale of scientific curiosity and collaboration. In the early 20th century, scientists suspected that something inside our cells carried genetic information, but they weren't sure what it was. It wasn't until 1953 that James Watson and Francis Crick, building on the work of Rosalind Franklin and Maurice Wilkins, unveiled the double helix structure of DNA. This breakthrough changed biology forever, revealing the molecule responsible for inheritance and the blueprint of life. Their discovery set the stage for modern genetics and biotechnology. DNA is a long molecule made up of smaller units called nucleotides. Each nucleotide contains a sugar, a phosphate group, and a nitrogenous base. These components link together to form a chain, and two such chains twist around each other to create the famous double helix shape. DNA is found in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotes. Its main job is to store genetic information, which is passed from one generation to the next, ensuring the continuity of life. Let's break down the building blocks of DNA. Nucleotides. Each nucleotide consists of three parts: a deoxyribose sugar, a phosphate group, and a nitrogenous base. The sugar and phosphate form the backbone of the DNA strand, while the bases stick out like rungs on a ladder. There are four types of nitrogenous bases: adenine, thymine, cytosine, and guanine. The specific order of these bases encodes genetic information, much like letters in a sentence. Double helix is one of the most iconic images in science. Imagine a twisted ladder where the sides are made of sugar and phosphate and the rungs are pairs of nitrogenous bases. This structure is not just beautiful, it's functional. The twisting allows DNA to be packed tightly inside the cell nucleus, and the specific pairing of bases ensures accurate copying during cell division. The double helix is truly a marvel of nature's engineering. DNA's structure relies on the precise pairing of nitrogenous bases. Adenine always pairs with thymine, and cytosine always pairs with guanine. These pairs are held together by hydrogen bonds, creating the rungs of the DNA ladder. This complementary base pairing is crucial for DNA replication and for maintaining the integrity of genetic information. It's like a code that ensures the right message is passed along every time a cell divides. Backbone of DNA is formed by alternating sugar and phosphate groups. This sturdy structure provides support and protection for the delicate genetic code carried by the nitrogenous bases. The backbone's arrangement also gives DNA its directionality, which is important for processes like replication and transcription. Without this strong framework, DNA wouldn't be able to maintain its shape or function properly. Within the long strands of DNA are segments called genes. Each gene contains instructions for making a specific protein, which carries out vital tasks in the body. DNA is organized into structures called chromosomes, which ensure that genetic information is accurately distributed during cell division. Humans have 23 pairs of chromosomes, each packed with thousands of genes that determine everything from eye color to metabolism. One of DNA's most important functions is replication. Before a cell divides, it must make an exact copy of its DNA so that each new cell receives the same genetic information. The double helix unwinds and each strand serves as a template for a new complementary strand. Enzymes help add the correct nucleotides, ensuring accuracy. This process is essential for growth, repair, and reproduction in all living things. DNA replication wouldn't be possible without the help of specialized enzymes. Helicase unwinds the double helix, while DNA polymerase adds new nucleotides to each template strand. Other enzymes help proofread and repair mistakes, ensuring the new DNA is an exact copy. This teamwork among enzymes keeps our genetic information safe and accurate, generation after generation. DNA's information isn't used directly to make proteins. Instead, it's first transcribed into a molecule called RNA. During transcription, a section of DNA unwinds, and RNA polymerase builds a complementary RNA strand. This RNA, called messenger RNA (mRNA), carries the genetic code from the nucleus to the ribosome where proteins are made. Transcription is the first step in turning genetic information into action. Once mRNA reaches the ribosome, the process of translation begins. Here, the genetic code carried by mRNA is read in sets of three bases called codons. Each codon specifies a particular amino acid, which is added to a growing protein chain. Transfer RNA tRNA brings the correct amino acids, and the ribosome links them together. This is how DNA's instructions are turned into the proteins that build and run our bodies. Genetic code is the set of rules by which information in DNA is translated into proteins. Each three-base codon in mRNA corresponds to one of 20 amino acids or a stop signal. This code is nearly universal among all living things, highlighting our shared evolutionary history. The genetic code ensures that the right proteins are made at the right time, keeping cells functioning smoothly. Sometimes mistakes occur when DNA is copied. These changes, called mutations, can alter the genetic code. Some mutations have no effect, while others can lead to diseases or beneficial traits. Mutations are a source of genetic diversity, driving evolution and adaptation. Cells have repair mechanisms to fix most errors, but occasional changes are part of life's ongoing story. Maintain genetic stability. Cells have sophisticated DNA repair systems. These mechanisms detect and correct errors that occur during replication or from environmental damage. Enzymes scan the DNA, remove faulty sections, and fill in the correct nucleotides. Without these repair processes, mutations would accumulate, leading to malfunction and disease. DNA repair is essential for healthy living. DNA is the molecule of inheritance. When organisms reproduce, they pass copies of their DNA to their offspring. This transfer of genetic information ensures that traits are inherited from one generation to the next. The combination of DNA from both parents creates genetic variation, which is the foundation of diversity within a species. Heredity is how life's instructions are handed down through time. Information stored in DNA is called the genotype. This genetic blueprint interacts with the environment to produce an organism's observable traits known as the phenotype. For example, your genes may determine your potential height, but nutrition and health also play a role. The interplay between genotype and phenotype shapes every living thing, making each individual unique. DNA's uniqueness makes it a powerful tool in forensic science. By analyzing DNA samples from crime scenes, scientists can identify individuals with incredible accuracy. Techniques like DNA fingerprinting compare specific regions of DNA to match suspects or victims. This technology has revolutionized criminal investigations, helping to solve cases and exonerate the innocent. Genetic engineering is the process of altering an organism's DNA to achieve desired traits. Scientists can add, remove, or modify genes to create crops that resist pests, produce medicines, or even cure genetic diseases. This technology holds great promise, but also raises ethical questions about how far we should go in editing life's blueprint. DNA sequencing is the process of determining the exact order of bases in a DNA molecule. This technology allows scientists to read the genetic code, identify mutations, and study how genes function. Sequencing has become faster and more affordable, opening new doors in medicine, ancestry research, and evolutionary biology. Human Genome Project was a landmark scientific effort to map all the genes in human DNA. Completed in 2003, it identified over 20,000 genes and provided a reference for studying genetic diseases in human evolution. This project has transformed biology and medicine, paving the way for personalized treatments and a deeper understanding of our genetic heritage. DNA is the engine of evolution. Over generations, mutations and genetic recombination create variation within populations. Natural selection acts on these differences, favoring traits that help organisms survive and reproduce. By comparing DNA sequences, scientists can trace evolutionary relationships and uncover the history of life on Earth. Epigenetics is the study of changes in gene expression that don't involve altering the DNA sequence itself. Environmental factors, lifestyle, and experiences can influence how genes are turned on or off. These changes can sometimes be passed to future generations, adding another layer of complexity to heredity and development. Our DNA plays a major role in health and disease. Some genetic variations can increase the risk of certain conditions, while others may offer protection. Understanding your genetic makeup can help predict health risks and guide personalized medicine, allowing treatments to be tailored to your unique genetic profile. Genetic testing analyzes your DNA to look for changes linked to specific diseases or traits. These tests can help diagnose genetic disorders, guide treatment decisions, and inform family planning. While genetic testing offers valuable insights, it also raises questions about privacy and how genetic information should be used. DNA testing can reveal information about your ancestry and family history. By comparing your DNA to reference populations, scientists can estimate where your ancestors came from and how you're related to others. This has made genealogy more accessible and has connected people with relatives they never knew existed. DNA technology has revolutionized agriculture. By understanding the genetic makeup of crops and livestock, scientists can breed varieties with better yields, disease resistance, and nutritional value. Genetic modification allows for the creation of plants that can withstand harsh conditions, helping to feed a growing global population. Medical research relies heavily on DNA. By studying genes, scientists can understand the causes of diseases, develop new treatments, and even design gene therapies to correct genetic defects. DNA-based diagnostics and personalized medicine are transforming healthcare, offering hope for previously untreatable conditions. DNA barcoding is a technique used to identify species using a short section of DNA. This method helps scientists catalog biodiversity, monitor ecosystems, and detect illegal trade in endangered species. DNA barcoding is a powerful tool for conservation and environmental protection. DNA isn't just for scientists, it's part of our daily lives. From paternity tests to ancestry kits, DNA technology is accessible to everyone. It's used in food safety, criminal justice, and even in tracking the spread of diseases. Understanding DNA empowers us to make informed choices about health, family, and society. With great power comes great responsibility. The ability to read and edit DNA raises important ethical questions. Who owns genetic information? How should it be used? Issues like genetic privacy, discrimination, and designer bobbies are hot topics in society. It's important to balance scientific progress with respect for individual rights. DNA databases store genetic information from millions of people. These databases are used in research, medicine, and law enforcement. While they offer benefits like solving crimes and advancing science, they also raise concerns about consent and data security. Managing DNA databases responsibly is crucial for public trust. Synthetic biology combines engineering and biology to design new DNA sequences and create artificial life forms. Scientists can build custom organisms for medicine, industry, and environmental cleanup. This cutting-edge field pushes the boundaries of what's possible, but also demands careful oversight and ethical consideration. Biotechnology harnesses the power of DNA to solve real-world problems. From producing insulin to cleaning up oil spills, DNA-based technologies are making a difference in health, agriculture, and the environment. The possibilities are vast, and ongoing research continues to unlock new applications. Some viruses use DNA as their genetic material. When they infect a host, they insert their DNA into the host cells, hijacking the machinery to make more viruses. Understanding viral DNA helps scientists develop vaccines and treatments and track the spread of infectious diseases. As we age, our DNA accumulates damage from environmental factors and normal cellular processes. Over time, this damage can affect how our cells function, contributing to aging and age-related diseases. Research into DNA repair and maintenance may one day help us slow down the aging process. Cancer often begins with mutations in DNA that disrupt normal cell growth and division. These changes can be caused by environmental factors, inherited genes, or random errors. Understanding the genetic basis of cancer has led to targeted therapies and improved diagnosis, offering hope to millions of patients. Personalized medicine uses your unique DNA to tailor treatments just for you. By analyzing genetic variations, doctors can choose medications and therapies that work best for your body. This approach increases effectiveness and reduces side effects, marking a new era in healthcare. Cloning involves creating a genetically identical copy of an organism using its DNA. This process has been used to clone animals like sheep and cows and holds potential for medicine and conservation. However, cloning also raises ethical and practical questions about identity and the limits of science. Stem cells are unique because they can develop into many different cell types. Their DNA contains all the instructions needed for specialization. By studying stem cell DNA, scientists hope to unlock new treatments for injuries, diseases, and even organ regeneration. Environmental factors like radiation, chemicals, and diet can affect our DNA. Some changes are temporary, while others can be passed to future generations. Studying how the environment interacts with DNA helps us understand disease risk and develop strategies for prevention. Our bodies are home to trillions of microbes, each with their own DNA. The collection of these genes is called the microbiome. Interactions between our DNA and the microbiome influence digestion, immunity, and even mood. Understanding this relationship is a new frontier in health research. Artificial intelligence is helping scientists analyze vast amounts of DNA data. AI can spot patterns, predict disease risks, and accelerate discoveries in genetics. This partnership between technology and biology is opening new possibilities for understanding and harnessing the power of DNA. Future of DNA research is bright and full of promise. Advances in technology are making it easier to read, edit, and understand DNA. From curing diseases to creating sustainable solutions, DNA science will continue to shape our world in ways we can only imagine. Staying informed and engaged with these developments is key to making the most of what DNA has to offer. Summary: DNA is the foundation of life, carrying the instructions that make each organism unique. Its structure and function are central to biology, medicine, and technology. From inheritance and health to innovation and ethics, DNA touches every aspect of our lives. As we continue to unlock its secrets, DNA will remain at the heart of scientific discovery and human progress.